So again, chapter 17 is all about biotech. Again, the best example, and the one we're going to look at mostly here, is doing DNA fingerprinting. Because again, remember, DNA fingerprinting is the best way to determine who a person is. Originally started out as just fingerprinting, but there could be some mistakes. DNA fingerprinting is much less mistake prone. So now there's basically three main categories of work. Y'all can probably list them all. What do you got? When would we use biotech, genetic engineering? What were three main categories? Where? Okay, so medicine. Okay. Okay, possibly crime. And? What do you normally hear the term GMO, or genetically modified organism?
Agriculture.
Agriculture. So yeah, mostly it's either medicine, agriculture, or crime. Okay? So, and it's hard to get away, specifically on the agriculture side, it is hard to get away from genetically modified organisms. Even if they say, oh, there are no GMOs in this crop, that's a lie. Unless you're pulling seeds or you're pulling animals that have never been in contact with human population, it's probably genetically modified. Specifically, more so on the plant side, because remember, plants, they've adapted themselves to be able to spread long distances, either through wind or through water or through being eaten and passed on. Those are all what we call seed dispersal adaptations. And plants have designed themselves to be able to spread. And so if you have a genetically modified corn or genetically modified soybean, and you're growing organic, non-genetically modified, there's a good chance that the pollen that you have contracted could possibly have come from originally genetically modified organism. Okay.
What was the third group?
So medicine, forensics, crime, and agriculture. And so here's the basic process of all of this. The basic process goes, essentially you obtain the plasmid from the bacteria. Plasmids, remember, plasmids are small circular forms of the bacteria, of the bacterial DNA. It is not the entire DNA, but it's a small section of that DNA. You then put a foreign gene into there. Okay, that gene, whatever the goal is, that's the gene you're going to put into the plasmid. You're then going to put that bacteria into a growing medium, and then that bacteria will then replicate and grow, because remember, bacteria are identical. So if you have one bacteria that you change, the next bacteria that's grown, the next bacteria that's grown, the next bacteria that's grown, through binary fission, it's going to be exactly the same, because it's all asexually reproduced. And essentially that's the deal. And so the bacteria grow, and so if you're using bacteria to produce insulin, which is a genetically modified, genetically engineered device, you're going to allow the bacteria to grow and produce the insulin, and then that insulin is then going to be collected and then used for diabetic people and other organisms. A couple little terms you need to know. First off, restriction enzymes. We will do an activity today about restriction enzymes, but essentially restriction enzymes are scissors. Restriction enzymes equals scissors. And so, like I said, you're going to take a specific section of gene, or you're going to put a specific section of gene into the DNA. And so for this to be inserted or for this to be pulled out, you have to cut parts of the DNA apart. So restriction enzymes allow for that. There's two different ways you can cut. Either, and we're going to look at this here in a little bit, we're basically going to have two different types, either sticky ends or blunt ends. And the restriction enzymes will determine which one of those are. The sticky ends, that's the one that's highlighted here, is the more important version of it, just because they do make for a better bond. Okay. The vector, it's not like vector like communicating crime with magnitude and direction. It's more vector as in being able to, how does it transfer? Oh, that was a good one. That was a good joke. No, but what vectors are for genetic engineering is that's the transport mechanism. So when we look at genetically modified organisms, how are we going to get that gene into the new organism? Okay, and it could be as minor as just using a bacteria or a virus to implement that DNA into the virus. A vector also, when we look at epidemics or pandemics, what was the vector? What's passed along? So, like you look at the Spanish flu of 1918, the vector itself was the transfer between the close proximity and actually swine, the proximity of the pigs in the area, and was able to jump from bird flu to swine flu, and then from swine to actually get to humans. So the vector would be whatever it is that's transporting the gene. And then the host organism, well, that's what's doing the actual the actual growing of that protein. And most of the time what we're looking at is the protein itself. Because remember, genes code for proteins. It's the central dogma. The DNA makes RNA, RNA makes proteins, and so if we can insert a specific section of DNA, then we can make new different types of genes. Main thing of these are all gel electrophoresis. Okay, this is that DNA fingerprint. This is the Maury Povich, you are not the father. Those kind of things. All right, and we're going to separate its size by electricity. So DNA itself is actually negatively charged, and so what you're going to do is up here on the very top. A little awesome picture going here. Ready? Here's my awesome picture. So at the very top, you're actually going to connect a negatively charged electrode and a positively charged electrode on the other side. And you're going to say, ready, set, go. And the DNA is going to transfer its way down this way. Okay. Now, I think I pointed out this last time, but what is the difference between these that are up here at the top and these that are down here on the bottom? Oh, just look at. Actually, we'll match. We'll actually look left to right as far as matching. Yeah. So what is the difference between those that are on the very top? Look at the numbers. What do the numbers do?
They get smaller.
They're descending, right? And so they're getting smaller. So what that actually is those are the number of base pairs that each of these segments have. So if you'll look up here at the very top, like this guy right here, okay, that segment right there is about just under 1,300 or just over 1,300 base pairs. That's 1,300 A's, T's, G's, and C's. Okay, but if you go down here to this segment right here, that's just under 200 base pairs, 200 A's, T's, G's, and C's. So essentially, as you go down, the smaller fragments are going further. Did I give you all an example about the race? No. Okay, I forgot about all of them. So anyways, here's this idea of a race. Okay, so we're going to have a race. Now, who in here feels they are the least athletic in this entire class? All right, so we're going to take the two least athletic people in this class, and I bet you that we can. Who thinks the most athletic here? So we've got Jordan back. Jordan thinks he's the most athletic. So here's the thing, Jordan. Do you think you could beat Brooke in a race? Like by no problems at all, right? All right. Here's what we're gonna do. We're gonna see who can run the furthest. We're gonna see who can run the furthest in 15 seconds. Okay. So we're gonna see who can run the furthest in 15 seconds. Now, so obviously Jordan we're gonna go with the idea, just even though Jordan's probably got a little bigger head than most of you, right? But let's say that Jordan can actually beat Brooke. Let's just say that. Let's say we're into this imaginary unicorn pony rainbow world. Right? So Jordan and Brooke are going to say, ready, set, go, and click the timer. And I'm going to give them 15 seconds, and they're going to see how far they can run the football field. All right. So Jordan books it out, and he's like, whatever it is, and Brooks. Okay, now, but I say, wait. Let's make this a little more realistic. So what I do is actually I take Devin and Jordan and Maddie and Minnie. I tie all their legs together. So all four of them are going to be all tied together, and then we all race together. So I'm going to tie the three guys in the back, and we're going to tie this whole row right here, the third row. We're going to tie all their legs together.
I think we're good guys. There's the least amount.
We're going to be groups of five right there, and another group of five right here. Okay. Now Brooke's going to run by herself.
Should win then.
All right. Who's going to win the race now? Guaranteed. Most likely guaranteed.
Still me.
Possibly Jordan. Possibly still no. All right. No. Hopefully the idea is that Brooke would win because she is by herself, right? She's the smallest. She doesn't have a whole lot of just random stuff hanging on to her, right? Okay. Now, let's say for instance, let's actually make this a little more realistic. Let's make this a little more realistic. And instead of it just being a nice, flat, even football field, I'm going to take the hurdles. I'm actually going to put the hurdles in the middle of the football field. All right? And so the hurdles themselves, so if here's your football field, right, we're going to place the hurdles kind of all throughout here. So there's little passages, but you have to find those passages to be able to get down to the bottom. Okay? Even more correctly, the person that's by themselves, the smaller unit of people, right, is going to be able to weave their way through the hurdles and find their way to the end much quicker, correct? Do we agree with that idea? No? Yes?
I think
Okay.
two heads are better.
That's basically what's going on here, okay? So these right here, again, this is about 1,000 base pairs. Okay, so right here, that's about 1,000 base pairs. So anyways, right here, that would be like that entire third row. There's about 1,000 base pairs all linked together. And those 1,000 base pairs had to fight their way through this gel electrophoresis. Okay. Now, this gel we're going to use is called agarose gel, and it's a lot like gelatin. It has solid chunks in it, just like your gelatin has solid chunks. Not solid chunks you can feel or taste, but remember we're talking about DNA here, teeny tiny bits of molecules. Okay, and those molecules, they had to find their way through the thickness of this gel, and so things that are very, very small are going to be able to go through much quicker than things that are very large. Do you agree with that idea? And so that's what we're going to look at here. So those restriction enzymes, what do restriction enzymes do? What did I say? Restriction enzymes equal? Scissors. So they're going to cut up this DNA at very specific locations. Now, we've got, in the thousands of base pairs in your body. Okay, so you've got thousands and thousands and thousands of base pairs in your body. Do you think all of us at some point within our DNA most likely has an AATTCC? We go with that idea?
Yeah.
Most likely, right? Six base pairs within 6,000 base pairs. Most likely you probably have an AATTCC. Okay? Now, Luke's AATTC might be on base pair number 126. Alan's AATTC might be on base pair 1055. Victor's base pair might be, wherever it is. And wherever that specific base pair is where that restriction enzyme is going to cut. So that's how we end up with smaller and big chunks and small chunks, is because how far along that restriction enzyme cuts is how big that base pair is going to be. Does that make sense? Because what this restriction enzyme is doing is it's looking for a specific location to cut. And it's looking for that specific code to cut. Okay?
What's wrong? Learn.
No, you're not.
That is biology. It's fine.
Okay, so that's basically the idea of gel electrophoresis. But there's different versions of gel electrophoresis. The first one is Sanger method. And what these are all different forms of the same test. But what are we looking for? So if we're looking for a specific sequence, let's say that we want to find what is the specific sequence for the human growth factor gene. Okay? We're going to use what's called the Sanger method. So again, the Sanger method is going to determine a specific sequence. And the way they do that is by using radioactive tracking with the specific nucleotides. Okay? And this is the way they had to do their very first genome. Before they had computers and everything, they actually had to take these little charts right here and say, okay, at base pair number 126, it's a T. At base pair number 242, it's a C. At base pair number 345, it's a C. And they had to do letter by letter by letter with a method just like this. Okay? So again, this is the way you would be able to figure out what the sequence is of a specific gene. Okay, so the Sanger method. Next one is PCR. PCR machines would be awesome to have in school, but they're like $5,000. So it's not going to happen. But essentially what we do is, let's say we have we go to a crime scene. And on that crime scene, we've got the culprit sneezed in the crime scene, and there's a little glob of mucus over in the corner, right? So we swab it. Now in that mucus, it's kind of dried out, but there's still a little moisture left, so some of the DNA is still intact. It's a very, very small amount of DNA. What the PCR does is it actually goes through replication, time after time after time after time, very, very quickly, to where after a polymerase chain reaction goes through, it'll actually have a large chunk of DNA from a very small amount. So the results of the fingerprint is actually much more viable than if it didn't have the PCR machine. But again, basically all it does is it just copies that specific section of DNA. So you would just grab that small section, and it makes copy after copy. So that one copy makes 2, those 2 make 4, those 4 make 8, then on that 4. Okay? And again, since it's just DNA, it can actually copy very, very quickly. So you can get, instead of it being a 120 base paired section of DNA, you can get to the 5,000 base pair DNA, within 2 hours or something like that. So it's very quick. There's 2 different forms. It's called the southern blot and the northern blot. And essentially, it's just basically what are we looking for on these 2, and what the technique is. Southern blot is where we're determining the sequence is in a specific organism. So let's say we know that the gene sequence, if you have a repeat of ATA 20 times in a mouse, that mouse is going to contract skin cancer 85% of the time. Okay, so you have in a mouse, and then that mouse's genome is ATA, and it's 20 times repeat. Okay. What we want to see is if we sequence a human genome, and they've got the same ATA 20 times, do they also contract skin cancer as well? And so what you do is you use a southern blot. It's actually a combination of gel electrophoresis as well as X-ray technology. Essentially, you run the gel electrophoresis, and in the process, you actually blot with a X-ray film. And that X-ray film then is able to form this blot right here, okay, what we call a southern blot. And again, what we're doing is we're comparing to see if one DNA sequence is going to be in both organisms. So dogs and humans, or mice and humans. Most of the time, again, this would be what we would use in research. If we find a gene that seems plausible for a problem we're working on, this would be a format of gel electrophoresis you're going to use. Okay, so again, this is a southern blot, and it uses X-ray technology to determine if a sequence is in both organisms. Whereas the northern blot looks at if a gene is made into mRNA. Okay, so let's say you have a diabetic, and we're trying to find a new drug for this diabetic. We're going to figure out if that gene that codes for insulin is being made in mRNA. Because if we can fix that mRNA, or if we can block that mRNA from continuing on, then we can actually fix the diabetes. So this one is not going to, this one again is going to use the same kind of X-ray technology. What we're doing is we're going to actually snippet a small section of that gene. Okay, we're going to run it through. We're actually going to look at, instead of the DNA, we're actually looking at the RNA. So northern blot, we're looking at RNA. Southern blot, we're actually looking at DNA. Now the RFLP, which is the restriction fragment length polymorphism, this is where you're looking for specific banding. So let's say you're looking for, well, where is the gene for sickle cell anemia? So if we're looking for that specific gene, we're going to use an RFLP, which is the restriction fragment length polymorphism. And these different alleles, if you have, remember, if you got sickle cell anemia, and let's say you've got the allele, you've got dominant, homozygous dominant, and you don't have sickle cell anemia. But if you've got this, and you're considered a carrier, but if you have this, little c, you have sickle cell. Okay? So what we're going to do is that each of these alleles are going to show a different band. So if we're looking for specific banding, or we're looking for specific alleles within an organism, say if you're doing genetic testing, and you want to know what is the likelihood of your kid having cystic fibrosis. Well, to have cystic fibrosis, you can be a carrier, you don't actually have the disease. So what we're going to do is you can do an RFLP test to determine if you are a carrier, and also if your spouse is a carrier as well. Because if both of you are a carrier, what's the chances of your kid having cystic fibrosis?
Very good.
What is it?
Wait, if you're a carrier...
If you're a carrier and your wife is a carrier.
No, it's like 50-50. No, 3
It's like straight 100.
No. What is it, Dusty? No.
No, 25.
25%.
Wait,
Okay, again, if you're a carrier,
If you're both heterozygous...
yeah, if you're both heterozygous. So we cross,
the only one that is for sure.
because the only one that would have the bottom right? Okay, so 25%, your kid's gonna have cystic fibrosis. So this is something that you want to do if you have that in your history of your family. The other one is in vitro mutagenesis. What does genesis mean? The beginning, right? So creation. Genesis, when you see Genesis, it actually means creation. Okay, because Genesis is the story of creation. And so mutagenesis is actually the changing, but you're actually changing it into a new creation. Okay? So this is actually in the specific organism. What we've looked at before is we've looked at, let's take a gene, and let's pull that gene out and let's put it in an organism. This one, we're taking the gene out and we're putting it back in the same organism after we mutate it, after we change it. Okay? So same idea, but again, we're going to cut out a piece of gene, we're going to mutate it somehow, we're going to chemically modify it, and we're going to put that gene back into the organism. Okay, so what we're doing is we're seeing, well, what is the effect of, if we change this gene, what's the effect on the long run process? This is how they're able to figure out things like cures for cancer, or things like cures for autism, or things like cures for Alzheimer's and those kind of things. Okay, is by saying, well, if this gene is what's causing autism, then if we adjust that gene and we put that gene back into that organism, does that organism still have autism? Or does that organism still have heart disease? Or whatever it is. Okay? Because this is then when you take this and you put it into like things like gene therapy. Because if we can give someone that has sickle cell anemia transfusions, like I have a friend of mine that lives in Philly, and he's had 2 years out on in Philadelphia. But anyways, he's like 2 or 3 years out on what he calls transfusions. He has a very bad case of I just went blank. But anyways, it's when he's allergic to gluten, celiac disease. There you go. He has a really bad case of celiac disease, and so what he's been doing is actually been getting transfusions, and to basically to counteract the mutation he has against gluten. And so because of that, he actually hasn't had an outbreak in over 2 years because of this. So what he's doing is he's actually changing his genes. He's fixing this problem by changing his DNA, which is kind of cool.
That's crazy.
Up here, this was the Human Genome Project. Y'all read a lot about this in the genome book. But again, remember, it worked much quicker. Yeah, you read a strange book, right? Yeah, I know. You ready to read another one? No? Okay.
Sure.
But the goal here was to basically map the entire human genome, figure out what diseases are specific locations, and then also analyze these genomes and relate it to the genomes of other species. So this is another piece where we were able to find out evidence of evolution was because of the Human Genome Project. Because of the Human Genome Project, we could say, well, this DNA is here, and this sequence is for determining what side is head and which side is tail. It's called the Hox gene. And the Hox gene determines which direction basically to grow. So if you adjust the Hox gene on fruit flies, it's the same Hox gene on humans, that you can adjust basically the way that things grow. So if you adjust it, the wings will actually grow backwards, as far as I know.
So they'll fail.
They don't fly because they're backwards. Because what we did is we adjusted that gene and said instead of here's the head and here's the tail, we said, well, here's the tail and here's the head. Okay?
I thought what you meant was instead of flying before they go.
Backwards? No, just doing work. But it did go much quicker. It was finished in 2013. Yep. The DNA technology medicine, so here again, this is one of the ones, so again, we go into things like gene therapy, where we're actually adjusting the DNA. We're creating vaccines as well as other drugs to affect these ideas. Okay? And then finally, the idea of DNA and fingerprint forensics, and then agriculture. These are considered solid evidence in courts, which is good, right? But also, that's where the whole well, it wasn't my DNA, I was actually there, right? But also the good thing is that old cases, they're actually now pulling these old cases back, and they're saying, well, here's more evidence, and they look at the DNA and they're actually acquitting people for things they've sat in prison for 10, 15 years. So again, you would use things like PCR to pull the small samples, like the little mucus that's over on the side from someone sneezing. And you would then run an RFLP to make those DNA fragments. What specific segments are related? So again, this is the idea of if you run that gel electrophoresis, and here's the blood, and here's suspect 1, and here's suspect 2, then obviously who's the criminal? Here's 1, and here's 2, right? The second person. Because it's very, very clear, because again, the restriction enzymes, those scissors, are going to cut at specific locations. They're going to cut it just at AATTC, or whatever it's looking at. And if AATTC is cut on base pair number 26, it's going to cut on base pair number 26 for the criminal.
Yeah, how'd you
Evolution talk.
Anyways, and then finally, this idea for agriculture. So again, the forensics, medicine, and agriculture. These are all ways that we've increased agriculture production through genetically modification. Okay? But all of these, again, all of these have trade-offs. All of these have benefits, but they all have things that we can see as being an issue. Okay? Like the salmon. We actually make genetically modified salmon because the salmon no longer run the rivers that they're supposed to run. Okay? Because the rivers that they're supposed to run have actually been blocked off by dams. But the problem is that those genetically modified salmon don't have the same resistance that wild salmon will have. So they're like, well, we'll just put the genetically modified salmon in. Well, they don't have the resistance, and so they get sicker, and they don't make their journey as well as they don't understand the idea of wild. And so they'll go up, and they actually are eaten much more quicker by predators than those that are wild salmon, because wild salmon have been bred into them how to basically not go towards the predators. Because remember, predators, one of the ways that they actually are easier to eat is because of camouflage, trickery, and that kind of stuff. The one I always think about is this right here. This is bug-resistant crop. You can have tomatoes that will kill a bug, will kill a grasshopper when it eats that tomato plant, because it eats it and it basically falls over. It's got pesticide in the tomato itself. And then you go and you buy that tomato and you eat it. They say, well, it's safe for you. Well, there's a little bit of iffiness there because it doesn't. Yeah, it kills a bug. It's not going to hurt you. So, anyways, these are the kind of things that you kind of have to look at, especially now because GMOs are becoming so frequent that you have to kind of like be a informed consumer. I guess is what I'm trying to say. So.
Yeah, but then there's a lot of stupid